So before I begin, I would like to start by acknowledging the summit organization for this invitation and the opportunity to participate in the online summit of Science, Nutrition, and Public Health 2021. Today, I will be presenting a part of a broader project entitled Quantitative Risk Assessment using challenge testing and predictive modeling to assess Listeria monocytogenes in a ready-to-eat food. So, as we all know, today we live non-stop stressful and more demanding lifestyles, and this influences food consumption patterns. And one of the major trends is the growing preference for convenience foods that are easy and practical, such as ready-to-eat foods. These are associated to long refrigerated shelf lives and do not require a heat treatment prior to consumption. Um, on the other hand, Listeria monocytogenes is a gram-positive bacteria, and unlike the majority of foodborne pathogens, Listeria monocytogenes is able to grow at refrigeration temperatures. This potential pathogen ability to adapt to very different and harsh conditions, such as a wide range of pH, temperature, water activity, osmolarity, grants the colonization of food environments such as processing plants, retail establishments, restaurants, household kitchens, and so on. The subsequent resistance of Listeria monocytogenes in the food processing environment can be linked to some known traits of this pathogen, like biofilm formation and tolerance to disinfectants, commonly used in the food industry, making the control of this pathogen in the food industry a significant challenge. These characteristics of Listeria monocytogenes combined with ready-to-eat foods allow the potential pathogen to multiply in foods during shelf life, even if Listeria monocytogenes is present in very low initial numbers, potentially threatening consumers' health. In fact, Listeria monocytogenes is responsible for many cases and outbreaks of foodborne disease through related to ready-to-eat foods, including dairy products, processed meat, salads, and others. The disease caused by Listeria monocytogenes is listeriosis, a rare but very severe human disease. Listeriosis is almost entirely transmitted through the ingestion of contaminated foods, mainly ready-to-eat foods. Listeriosis has a high fatality rate. As you can see in this slide, it reaches or peaks at almost 20% of fatality rate. And this is the highest of any foodborne pathogen, even when antibiotic therapy is used. This is especially true for high-risk populations such as pregnant women, newborns, elderly, and immunocompromised people. According to the latest European report on foodborne disease, as can be seen in this image, on the right side of the slide, there has been a significant increasing trend in listeriosis cases in Europe, and in Portugal, the scenario is very similar. However, the growing trend in listeriosis cases has not only been observed in Europe, but also in other countries such as South Africa, where the deadliest listeriosis outbreak in history has been recorded. A ready-to-eat processed meat product was identified as the source of the outbreak. Due to the importance of this pathogen in ready-to-eat foods, there are regulations, mandatory regulations in Europe, and according to Commission Regulation number 2773, there can be two kinds of food products depending on their ability to support the growth of Listeria monocytogenes. For foods with these physical chemical characteristics and shelf life inferior to 5 days, they are considered not able to support the growth of Listeria monocytogenes. However, for those food products that do not present these characteristics, they are ready-to-eat foods able to support the growth of Listeria monocytogenes. In these cases, the food business operator is responsible to evidence that the limit of 100 colony forming units per gram of food will not be exceeded during shelf life. Of course, that the food business operator should base his decision on several studies, including food challenge testing, predictive microbiology, and risk assessment. Especially food challenge testing, which may be the base for the other two, consists of artificially inoculated food products and then studying the microorganism's behavior and growth during shelf life. There are a few guidance documents on how to conduct these studies. Therefore, it is of utmost importance to understand the behavior of Listeria monocytogenes in ready-to-eat foods. For that, this study aims to develop a challenge test using a ready-to-eat chicken salad, having as an outcome two important tools: predictive growth models and quantitative microbial risk assessment to estimate listeriosis cases per year linked to ready-to-eat chicken salad consumption. The choice of the sample to be analyzed, the ready-to-eat chicken salad, was not random in our study because this salad is produced in an industrial unit located in the metropolitan region of Lisbon and has a history of environmental persistent contamination by Listeria monocytogenes within its premises. This salad is prepared manually in a production line located in a temperature-controlled room around 10 to 12 degrees Celsius, and the ingredients are placed in a polyester salad bowl, as you can see, and after production, salads are stored for 6 days at 5 degrees Celsius. So, in terms of workflow, our work began with the development of a challenge test in which the physical, chemical, and microbiological characteristics of the salads were studied. Listeria monocytogenes growth was studied to allow for the development of growth models, which were used to perform a quantitative microbial risk assessment on these salads. The first thing that had to be done in the challenge task was actually choosing the strains to be inoculated in the salads, and to account for variation, a mix of three reference strains was used. These strains belong to serogroups 2A, 2B, and 4B, which account for more than 90% of foodborne listeriosis. In order to recreate an adaptation to the refrigerated food-producing environment, which was a major challenge for us, the three selected strains were firstly incubated separately in BHI for 4 days at 12 degrees. Cells were then centrifuged, washed, and finally they were resuspended, then mixed together and diluted to obtain a suspension containing approximately 4 log CFU per milliliter, which was used to inoculate the cells, as you can see in the photo, in the photograph. Um, with the help of a pipette. Samples were then incubated at 4, 12, and 16 degrees Celsius for 8 days, and three independent replicates were performed for each temperature. Besides the inoculated samples, blank samples were also considered. In terms of food analysis, physical-chemical determinations included pH and AW, while microbiological analysis included as hygiene indicators Enterobacteriaceae and total mesophilic counts, and Listeria monocytogenes detection and quantification were also performed. Below on the slide, you can find the sampling time points. In terms of predictive growth models, the primary growth model described by Barani and Roberts was fitted into the obtained microbiological data for the estimation of the Listeria monocytogenes growth kinetic parameters like lag time, maximum growth rate, and more. For the development of the secondary model for maximum specific growth rate, a square root model proposed by Rutkowski was used. In this study, a quantitative microbiological risk assessment was also performed. The model estimated the annual number of listeriosis cases linked to the consumption of these ready-to-eat salads, focusing on the home storage phase, considering consumption by different groups of the population. The model was built using R programming language and considered three main modules. In the exposure, as you can see on the left of the slide, in the exposure assessment, we assessed prevalence and concentration of Listeria monocytogenes and developed the predictive growth models. In the observed characterization, we established the serving size and determined the dose response model. Finally, the risk characterization module combined the first two steps to characterize the risk of listeriosis by predicting annual number of listeriosis cases using a Monte Carlo stochastic model. Sensitivity analysis was also performed to identify the factors that contribute the most to risk augmentation in the consumption of ready-to-eat salads. The selected factors were initial concentration of the pathogen, portion size, storage temperature, and time. On to results. So pH and AW measurements confirm the ready-to-eat salad as a food able to grow, to support the growth of Listeria monocytogenes. The growth curves were obtained by fitting Barani and Roberts model on experimental data. Listeria monocytogenes have the ability to survive and grow in this type of food or in this type of ready-to-eat salad at all of the assessed storage temperatures. However, initial and final concentrations are very similar among storage temperatures. During challenge testing, and Listeria monocytogenes growth behavior was different because of that. As the storage temperature increased, lag time decreased. In contrast, the maximum specific growth rate increased. Significant differences were detected in the maximum specific growth rate for the three temperatures, confirming that temperature influences the pathogen growth. Data obtained in the primary model of growth was used to elaborate the secondary model according to Rutkowski, which allowed to predict the maximum specific growth rate as a function of temperature by using this the expression shown in the slide. Considering quantitative microbial risk assessment results, the average number of listeriosis cases per year linked to the consumption of these ready-to-eat salads was 8.5 to 10 to the fifth. -5, I'm sorry. Moreover, when comparing the estimated number of listeriosis cases in low and high risk subpopulations represented by the both peaks we see on the graph, it is possible to observe that high-risk population has a 4 times higher risk of listeriosis. Thus, particular preventive tips should be given to high-risk populations. The sensitivity analysis allowed to determine that household storage temperature and the duration of storage were the most influential factors for the risk of listeriosis, derived from the consumption of ready-to-eat chicken salads. Initial Listeria monocytogenes concentration also had an average contribution to the risk of listeriosis, while the variability in the consumed portion size had no significant contribution to the total effect. Sensitivity analysis considering different time temperature scenarios indicated a very low median risk proportion, which was inferior to -7 log, even if the assessed ready-to-eat chicken salad was kept in abused storage conditions. On to the main conclusions of our study. pH and AW results confirm this study's ready-to-eat chicken salads as a food product able to support the growth of Listeria monocytogenes. Also, the results highlighted that temperature played a major role in the growth of Listeria monocytogenes in these salads. This work underlined the importance of strict temperature control from processing to consumption. Refrigerated temperatures must be maintained during transportation, distribution, storage, and handling in supermarkets and by consumers. The developed predictive growth models provided insights into predictive microbiology, which may be applied by the food industry and regulatory agencies to estimate the growth of Listeria monocytogenes in similar types of ready-to-eat foods. Quantitative microbial risk assessments and sensitive analysis indicated the need for strict adherence to time temperature recommendations as a risk management measure. This study may contribute to a better understanding and prediction of listeriosis cases by consumption of contaminated ready-to-eat products due to the rare development of microbiological risk assessment. I would like to acknowledge Rita for funding the project and my alma mater, FMG at University of Lisbon. Thank you so much.